Hello everyone, this is the Theoretical Doctor and welcome back to my channel. If you're new here, welcome. This is a series on the chapter Immunity of chapter 6 of the STPM Semester 2 Biology series. Check out the full playlist on my channel. If you want access to flashcards on this topic and other videos, do click the link below. It is available on my Pinterest and slides on this topic are available on my website. In this video, we will be starting on the human lymphatic system as well as the structure and roles of antibody, antigen, and the defense cells. In this video, we will be covering on the human lymphatic system and functions, antigen, epitope and macrophages, antibodies or immunoglobulins, classes of immunoglobulins, their structure and functions, and finally the differences between B lymphocytes and T lymphocytes. The human lymphatic system. The lymphatic system consists of a network of dead-end lymphatic capillaries and larger vessels, lymph nodes, the thymus gland, the spleen, and other satellite organs such as the tonsils, appendix, or the Peyer's patches in the small intestine. It is closely associated with the circulatory system as the lymphatic fluid is eventually returned to the blood via the thoracic duct and the right lymphatic duct. So the right lymphatic duct drains the right arm and the right half of the head and neck, and it drains into the right subclavian vein and the internal jugular vein. So this part here is the right subclavian vein, and this part over here is the internal jugular vein. The thoracic duct drains all other parts of the body, and it will drain into the left subclavian vein. The lymphatic fluid or lymph is clear and colorless fluid similar to blood plasma, except that it is low in proteins and does not have erythrocytes. It does not have red blood cell. It is formed as a result of the blood's high hydrostatic pressure. So the lymphatic capillaries, they are one cell thick, and one end of the lymphatic capillaries is closed, as you can see in the picture over here. This part here is a dead end, and the other end of the lymphatic ducts are equipped with valves to ensure a one-way flow of the lymph, so there's no backflow. The interstitial fluid presses against the lymphatic capillaries, allowing fluid to enter the lymphatic capillaries. So entry is one way only, and after it has entered, it cannot move out. There's no backflow, and the valves are there to prevent it. The pathway taken by the lymph from the lymphatic capillaries, it will enter the lymphatic duct. From the lymphatic duct, it will enter into the thoracic duct, and finally into the respective subclavian vein, either the right or the left, and eventually into the heart. So the lymph flows through lymph nodes found at specific intervals throughout the main duct, which contains lymphocytes and plays a role in immunity. So the interstitial fluid is formed when the blood moves under high pressure. So the blood will be pumped out from the aorta into the systemic circulation into the arteriole. So the blood will be coming under high pressure. As the blood moves under high pressure, eventually some of the interstitial fluid will move out. But not all the interstitial fluid can be taken up on the venial end. This is the venial end. So some of it enters into the lymphatic capillaries, and this will form the lymph, which will flow in the lymphatic capillaries, and the flow will be aided by the valves as well as the contraction of the skeletal muscles. Functions of the human lymphatic system. It is important in the removal of fluid and dissolved solutes from the interstitial spaces back into the circulatory system. It is also important in transport of fats and fat-soluble vitamins from the small intestine to the bloodstream. It is also important in the production of lymphocytes, which are responsible for the specific immune response, such as the humoral antibody response and also the cell-mediated immune response. So referring to the picture over here, the blood from the aorta flows to the systemic circulation of the body under high pressure, as I mentioned earlier, and interstitial fluid moves out of the capillaries. The water moves out of the capillaries. That one is known as the interstitial fluid. Now the interstitial fluid is absorbed back at the venial end, but not all of it. So some of it entered the lymphatic capillaries. As you can see here, the other end of the lymphatic capillaries are closed. closed. So the lymph will flow through the lymphatic capillaries, which has valves over here to prevent the backflow, and through the lymphatic system. So over there in the lymphatic system, there will be lymph node present, and this lymph node will be present throughout various parts of the body. So when the lymph passes by these lymph nodes, the toxins and pathogens will be engulfed by the phagocytes. So there will be lymphocytes and antibodies present over here, and the lymph will basically go through multiple lymph nodes like this one, and finally back into the thoracic duct or the right lymphatic duct, depending on which area is being drained, of course, and back into the blood circulation, as you can see over here. So it will go back into the blood circulation, and it will flow with the blood circulation back to the vena cava and back to the heart, and then it will be pumped into the lungs to be oxygenated and pumped out of the aorta again, and the whole cycle repeats itself. Antigen, epitope, and macrophages. So antigen are foreign molecules that are recognized by lymphocytes, which are stimulated to produce antibodies. So the antigens are basically the pathogens, the enemies. This is the antigen. Epitopes are antigenic determinants, which are found on the surface of the antigens. This one over here. So the autoantibodies, the immunoglobulin, they will recognize them. They will recognize the antigen through the epitope, and they will attach. So basically, epitopes are the uniform that the antigens wear. So that's how the immunoglobulin recognizes it's a pathogen. Macrophages, this one. So macrophages are cells which develop from monocytes. Monocytes are a type of white blood cell. Macrophages have surface receptors that bind molecules on foreign cells or substances. So these surface receptors are class 2 MHC. MHC stands for major histocompatibility complex molecules. So after the MHC molecules are synthesized in the macrophage, they bind to the fragments of ingested antigens and moves towards the plasma membrane. Basically, this macrophage will eat the antigen, and after they eat it, it will be phagocytosed, and a part of that antigen will be displayed outside at the plasma membrane through the class 2 MHC. So in this way, after it has displayed it, the macrophage becomes an antigen presenting cell. Presenting because it's showing it has taken up the antigen, and it's showing a part of the antigen outside of the macrophage, basically like holding a sign like here I've contained the enemy, so you can come and attack. Antibodies, or otherwise known as immunoglobulins. Antibodies or immunoglobulins are Y-shaped molecules made up of 4 polypeptide chains, 2 identical heavy chains, this one, they're identical, 2, and 2 identical light chains, okay? And they are linked by the disulfide bridges, the disulfide bond. So both heavy and light chains have a constant region that is identical for all antibodies of the same isotype. The variable region is designed by the B cell to specifically recognize an antigen. This part here, the one on top, like you can see like a knife like that, it's the variable region. Variable meaning it can change, it differs. This part over here is the constant region, meaning it will never change. So depending on the antigen, the variable part changes. For example, if this was an IgG, an immunoglobulin G, there are many immunoglobulin G in our body, but not all of these act on the same enemy. Not all of these act on the same antigen. Only those that fit the epitope of the antigen. So coming back here, this is the epitope. So if it doesn't fit, let's say, for example, this shape, right? This one is a bit different than this one. It doesn't fit. So it's not gonna attack that antigen. But let's say another epitope that fits over here, like lock and key, it fits. So that particular immunoglobulin or antibody will attack that antigen. So it depends on the variable region. This part here will act as the antigen binding site where the epitope of the antigen binds to. So some functions of the antibodies or the immunoglobulins: opsonization, to opsonize the bacteria, making them easier to be phagocytosed; neutralization, neutralize obviously, to neutralize toxins and viruses; complement activation, antibodies activate complement proteins, which leads to lysis of foreign cells. Lysis meaning to kill it. fluid. Precipitation. Soluble antigens bound to antibody molecules forming larger complexes, which then leads to precipitation. So precipitate, to let it condense, to go down. Agglutination, to clump together. Let's enter the classes of immunoglobulins, structure and functions. The first one, immunoglobulin M, or IgM. It is the first class of antibody produced when initially exposed to an antigen. It is responsible for the primary response, meaning they are first to appear in infection. However, these IgM do not cross the placental barrier, and they're mainly responsible for agglutination and involved in complement activation. And this is IgG, or immunoglobulin G. It is a small monomer. This one you can see it's a big one. This one is only one, which easily moves out of the blood vessels and into the tissue fluid. They are able to cross the placenta, thus conferring passive immunity to the fetus. They are most abundant and they promote opsonization, neutralization, and agglutination. They are responsible for starting the secondary response. So these two, IgM and IgG, are very important, especially which ones come first during an infection. During primary response, or when the person is newly infected, IgM comes first. That's why they are responsible for the primary response, meaning the first to appear in infection. Whereas after a few attacks or in secondary response, it is by IgG, this one. So how I remember this is mothers comes first. So IgM first, then IgG. So mother M, IgM. That's why IgG is involved in passive immunity. As you can see over here, passive immunity occurs when there is a reinfection. The body will recognize the antigen and react immediately. So the immune response will automatically be stimulated. Moving on, IgA, or immunoglobulin A. So they are fairly small molecule made up of two monomers, produced mainly by cells in the mucous membranes. Moving on is immunoglobulin A, or IgA. They are fairly small molecule made up of two monomers, as you can see over here, produced mainly by cells in the mucous membranes, also found in tears, perspiration, saliva, and colostrum. Colostrum is basically when a mother first gives birth, or sometimes before they give birth actually, some milk is being produced by the breast. So that first one that comes out before the mother continuously produces breast milk, that is the colostrum. Just a little bit about that. It prevents bacteria and viruses from attaching to the epithelial surfaces of the respiratory passages, alimentary canal, urinary, and reproductive tracts. Then we have IgD, or immunoglobulin D. It is mainly found on the cell surface membrane of B cells, where they function as antigen receptors. They cannot cross the placental barrier and are responsible for stimulating the differentiation of B cells into plasma cells and memory cells. So they help in signaling B cell activation. This is actually the humoral immune response, which I will talk in another video. IgE, the last one, or immunoglobulin E. It is found only in small amount compared to other antibodies. So they bind to mast cells, basophils, and stimulate them to produce histamines. They are responsible for allergic response and responsible for immunity to parasitic worms. Basically, when you see IgE, all you have to remember it's allergy and histamines. B lymphocytes versus T lymphocytes. So first one, B lymphocytes. B cells, or otherwise known as B lymphocytes, are bone marrow derived cells involved in humoral immunity, which is important in the production and secretion of antibodies. So if you see B lymphocyte, remember humoral immunity and antibody. So the function is to kill bacteria, and they will be able to differentiate to form into plasma cells, and memory B cells. Then we have T lymphocytes. So T cells or T lymphocytes are bone marrow derived but mature in the thymus. So if you see T, remember T for thymus. They are formed in the bone marrow, mature in the thymus. So they are involved in cell-mediated immunity. So T cells have surface protein CD4 or CD8. Okay, then depending. If CD4, they're called helper T cells. If CD8, they're called cytotoxic T cells. So helper T cells, they are able to activate humoral immune response as well as cell-mediated response. So they work both ways. They can activate this and they can also activate this, the cell-mediated pathway, which again I'll talk in another video. So their function is to inhibit intracellular bacteria and fungi. Then we have the cytotoxic T cells or CD8. They activate the cytotoxic T cells obviously, and they kill virus infected cells. But they're also important in terms of killing cancerous cells, but they're also dangerous because if a person were to receive organ transplant, these cells are the ones who are the culprit in actually rejecting the organ that's being transplanted. So more on that in the following videos. If you've reached this point, thank you very much. Hit the like button if you found it helpful, as it does help in reaching out to others as well. Do support my channel by pressing the subscribe button so I will know, and it gives me motivation to continue producing content like this in the future. And finally, share this video to those who might find this helpful. See you in the next one.